So my charge is to give you a deeper dive into the Humans in Nature program area, which I'm not going to do in the way that this slide would suggest I'm going to do. I'm not going to walk through the projects that we've run in the Humans in Nature program area. But I wanted to give you a sense of what's going on in the last decade, which is roughly period of time during which I've been one of the scholars as well as editor of the Hastings Center report. The funders are listed on the right. I should say that all this work in the Humans in Nature program area really is preceded by still earlier work that was led by Strawn Donnelly, a scholar and one-time president of the Hastings Center, actually the individual who hired me here at the Hastings Center. And it was listening to Strawn wrestling with questions about humans and nature that sort of inspired me to, almost on the sly, actually, write a proposal to the NEH and get the project at the top, the Ideal of Nature, going. That project looked especially at questions, sort of conceptual questions about the kind of relationship that we humans have to nature, and what kind of relationship we would like to have to nature. What should be the guiding ethic for the human relationship to nature? And I will, at the very end of this presentation, turn a little bit to that question. That kind of philosophical question then led to a few projects funded by Sloan about synthetic biology, and then we also began to broaden the scope of inquiry out so that we weren't just thinking about the guiding ideas in the human relationship to nature, but we were also thinking about more straightforward consequentialist kind of concerns. What are the impacts of the things we can do to nature? And how do we go about, what's the best way of assessing the impacts of these interventions in nature? With respect both to the benefits and risks for humans as well as those for the environment. And the project begun in 2014, Values and Impact Assessment, funded by the NSF, is focused on that. It takes some cases in synthetic biology as a sort of a case study. And then in the last this year, I've launched two new lines within the Humans in Nature area. One is a project on de-extinction, and the other is a project actually at the National Academy of Sciences on gene drives, and it is those that I'm going to talk about today. So I'm going to give you a dive into the Humans in Nature program area by exploring these last two issues, talking about what the technology is making possible in much the way that Professor Wu did with gene editing for humans, what the sort of emerging applications might be, and then I'll close by just laying out some of the questions that these applications raise, and we can have a conversation about that and about what to do with those questions. So, de-extinction. It is well known by now that we're in the middle of what is considered the sixth major extinction event. Elizabeth Kolbert came out with a book just last year. Was it called The Sixth Extinction? But she was picking up that term from others. We're in the midst of tremendous species loss, and it really probably began with human activities during the Pleistocene, during the Ice Age. Most of the megafauna of 10, 12, 15,000 years ago died out on each continent as humans moved to that continent. But we are now at a point where people are beginning to talk a little bit about de-extinction events, the possibility that we might be able to bring back at least some of these animals that in the last 10 to 12,000 years we have driven extinct. There are three broad categories of strategies for doing this. The first one has actually been around for quite a number of years. At least the idea for it has been around for quite a number of years, and it's not very high tech at all. It just consists of trying to backbreed animals to recover a missing animal. The Eurasian aurochs is the leading example of this. This is a representation of what the aurochs was. It was essentially, it was a cow, but it was a huge and extraordinarily athletic cow. So it was not like any cow you've ever seen. But all European cattle are descended from the aurochs. And in principle, there's little bits of the aurochs genome in various breeds, more of it in some breeds than in others. And so if you found the right specimen animals, those that have a phenotype, that is a body presentation, that seems most closely to resemble the original aurochs, as we understand it, and you cross them, then you could begin to isolate the original species. And you keep back crossing, working your way back, and in theory you could assemble, you could get back to the original phenotype pretty well. And you could also, if you can identify the genotype of the aurochs, you could use that as a kind of guide as well. And there are actually a couple of efforts underway in Europe to do this. One is called the Tauros project, and the bull in the little inset picture here is one that has been bred in the Tauros project. And there's another one called the Uruz project. Uruz is an old Germanic name for the aurochs. And it is funded by the True Nature, the intriguingly named True Nature Foundation. And the hope is to have herds of aurochs released into 10 or so nature preserves around Europe, and by 2020 or so. So not very long away. The original idea for trying to get back to the aurochs by backbreeding, incidentally, was devised during the Nazi era. There was interest among some of the Nazi leaders in trying to get back to sort of true nature. You can see how that connects with some of Nazi ideology. So it's an interesting thing for those who want to get back to true nature to wrestle with.
Is there any regulation, current regulation on that particular strategy?
None that I'm aware of at all, actually. There are probably European animal husbandry laws and so forth that bear on it, but there's no, that I'm aware of, no risk assessment being done on this. Partly because it doesn't rely on advanced molecular technologies. It's just breeding, it's very familiar. It's probably not subject to anything more than ordinary breeding technologies. This is a somewhat, in some ways, this is might be the best candidate actually for a de-extinction effort. I mean, we actually are probably somewhere along the way in getting to success with the Tauros project. But most people who are watching this would say that the eventual animal you'll get out of it will not be an exact representation of the original aurochs. It's going to be similar to it. It will be a functional equivalent of the original. So whether it's true nature or not, is still something. Yeah, Dan.
What is the point of research? Why do it at all? Who cares?
Because people want to recover true nature. I think that is really the primary rationale for it. There's also some hope that by reintroducing into Europe an animal that is a functional equivalent for the aurochs, that does the kind of thing that the original did, even if it doesn't quite count as the species, that there will be some ecological benefits from that. It will disperse seed and do some of the things that the original European ecosystems counted on the aurochs to do. But it's really about the human relationship to nature and some desire to see these things tromping around, and that's part of why I have big pictures of these things to sort of convey, to try and get across some of the excitement that the people who are doing it feel. So that's one strategy, and it would get you sort of a hybrid animal. Another strategy is to rely on cloning technology. The idea here is that you can recover a little bit of the actual tissue of the now extinct animal, one way or another, and you'd use that to create an animal that very closely resembles the lost thing. What you do is you, like say, you take a little bit of skin cell and you extract the nuclear material from it. Then you find a closely related species and you take an egg from the female of that species and extract the nuclear material from the egg. So you have a denucleated egg plus the nucleus of your missing animal, the genetic genome of your missing animal, and then you combine those two, that's cloning technology, somatic cell nuclear transfer, and you literally give it a little spark to get it to combine and begin to develop as an embryo. And if it does, then you implant it in the uterus of the animal from which you got the egg and try to bring it to term. And this has actually been done, it turns out, with the Pyrenean ibex, a subspecies of the Spanish ibex, known as the bucardo, that lived in a mountainous region of Aragon, near Barcelona. It was hunted to extinction, people thought, in the 19th century, and then in the late 20th century. This is incredible to me that this could happen in Spain. But in the 1990s in Spain, this little remnant population was discovered in the mountains. And they began tracking it. They radio-collared individuals, and by 1999 it had dwindled to a single female named Celia. And in 99, she was struck by a falling tree. Radio signals indicated she was not moving, she died, and they swooped in and collected some tissue, and in 2000 they managed to create a clone of Celia using a goat. And this is in the inset, it's a picture of the clone. The clone lasted for all of 7 minutes before passing away. The cloning effort didn't go very swimmingly, which is common for animal cloning. We're getting better at it, but particularly in 2000, there were hundreds of efforts to get an embryo going, hundreds of efforts to implant it, 154 successes, but almost all of those failed. A handful of pregnancies, most of those ended in miscarriages, and the one success was one kid for 7 minutes. So again, for me, one of the questions here is whether this actually counts as de-extinction. If you get one kid for 7 minutes, have you brought the species back? But unlike the backbreeding effort, you would have an animal that was genetically identical, except for mitochondrial DNA. You'd have an animal that was genetically identical to the lost animal. So you're not creating some sort of hybrid thing. You actually are getting back to something like the original. Yeah, no.
Greg, did they save the rest of the DNA for other later tries?
It is, and they are trying again. And depending on where you go, we could see an announcement any week now. Let's see what about a new effort. And there are some other animals for which this strategy is a candidate. One of them is the gastric brooding frog, a frog that from a biodiversity standpoint, it's very interesting. It is one of the, maybe the only animal known to bring its young to maturity in its stomach. The young are born as tadpoles and then eggs and then swallowed, and the stomach turns into a uterus and brings them to term, and then the froglets emerge in adult form, small but adult form from the mouth. So there's some feeling that this would be, for people who are interested, who care about biodiversity, there's some sense that it would be really, this is a major loss. This is a major loss to biodiversity, and so it would be attractive to have it back. There's a project ongoing to try and carry out cloning on this frog. It's called the Lazarus Project. These terms are very evocative and need a little bit of critical thinking, I think. The limitation, the chief limitation to this method is that you have to have very good tissue. Professor Wu was pointing out that DNA is pretty stable, but when it's in a cell out in the wild, it does degrade pretty quickly, and so there are very few animals for which you can use this, probably. It probably has to be something that you've collected, from which you've collected tissue and stored in a laboratory, stored in somewhere.
Is there a
Yeah.
DNA bank the way there's a seed bank?
There is.
There's, so species that are on the way to extinction or saving their DNA?
You could do that, and I think that there is an effort to do something like that. I should be careful about saying for sure. There are some scientists who feel that they can use this technique to bring back animals from the Ice Age. There's a Japanese scientist who has declared that he's going to bring back the woolly mammoth within the next few years doing this. By finding tissue that's frozen in the tundra somewhere, and you get something that's not been degraded at all, and maybe you could extract the nuclear material from it and put it into an enucleated elephant egg, and then bring it to term in an elephant. But some of the other scientists who are doing this work think that that's a pipe dream. It's never been happened. Eric?
So do you buy the assumption that mitochondrial DNA is irrelevant to the phenotype? Do you buy the claim that the environment of the cell into which the nucleus is inserted has nothing to do with the ultimate phenotype?
No, I don't say that it's irrelevant. And there's more, might well be much more than just the mitochondrial DNA that you'd have to control in order to make sure that it was really genetically identical to the parent. And Professor Wu was pointing in the direction of some of these mechanisms that we don't really well understand, maybe epigenetic phenomena or other things that are going sort of in and around the genetic material itself. So there is a real question whether if you brought back an animal through cloning, you'd have the original. But I suppose by my lights, it looks like although that material is not irrelevant, it may well play a small enough role that we could confidently say, yeah, we'll call this a Bucardo.
Can we agree we don't really know? We just really don't know. I'm familiar with the claims that might not be it's irrelevant. It seems to me we don't know.
We don't know. But we can, I think, we can take some
We can guess
guesses.
on either side.
So probably to bring back the mammoth, we need to rely on the third strategy, which does involve cloning, but adds to it some genetic tinkering. So in this strategy, rather than just sort of recovering the genetic material and then going through the cloning process, you have to start by creating the genetic material. The book pictured in the inset just came out, I think this year, and by a scientist who's been involved in recovering the stuff and then been involved in some of these efforts, and it's a wonderful read. The basic strategy here would rely on some of the genetic editing techniques that Professor Wu was pointing out, and might well involve CRISPR-based editing. It could involve some other things as well. George Church is centrally involved in all of this work, and is centrally involved in the effort actually to bring back the woolly mammoth. The idea would be that you can't synthesize all the genetic material de novo. You have to start with some sort of something given. So you'd start with something closely related. The most closely related animal for the woolly mammoth turns out to be the Asian elephant, probably more closely related to the mammoth than the African elephant is to the Asian elephant. So you could sequence the genome of the Asian elephant and sequence the genome of the woolly mammoth by looking at material you've collected from the tundra, and identify all the points of difference. If I recall, there are some 250,000 alleles that are different, and then go through and try to make changes where appropriate and as possible, and eventually turn the Asian elephant genome into a woolly mammoth genome. And then once you've achieved that, then you could implant it in an Asian elephant egg, bring it to term. You'd have all the problems of achieving pregnancy and producing an animal that survived, but in principle, you could eventually get a woolly mammoth out of this. Now, that's a lot of changes to make. So what's happening instead is that they're trying to identify a handful of key changes that make the Asian elephant functionally different from the woolly mammoth, make the mammoth functionally different from the Asian elephant. But if they can figure out what those changes are, for example, a gene that makes the blood a more efficient oxygen carrier at low temperatures, then you can affect those changes through genetic editing. And you can bring back an animal that will be a hybrid. It'll be a really hairy, cold-tolerant Asian elephant.